Good morning, this is Mr. Reynolds, and we need to study embryology today, okay? And embryology is um, the study of development of organisms, okay? And here's a look at something that you've seen before, sperm meets egg at fertilization, and the egg, the one egg gets fertilized, is known as the zygote. It gets fertilized. There's a wait period if everything matches up because we get half the chromosomes. We have 46, half the chromosomes from the sperm cell, half the chromosomes from the egg cell. Has everything matches up, and if it matches up just fine, then it gets the green light to go. And then this one cell divides into two, two divide into four, four into eight, eight into 16, 16 into 32, 32 into 64, 128, 256, 512, 1024, 2048, etc., 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 till we end up um, with with a ball of cells, basically a ball of cells. A blastula also has a little bit of hollow section developing inside of it. Okay, but basically it's a ball of cells. Okay, thousands of cells. All right, so this process of cells dividing is known as cleavage. So this is step one that we want to look at. Okay, and then we'll look at gastrulation in the forming of the gastrula, and these layers of cells is going to be critical. That's what I wanted us to see. Okay. A third step that we're somewhat familiar with already, even though you may not know it, organogenesis. We'll take a quick look at that also, okay? But I said we want to look at the cleavage, which will be pretty quick, and then the gastrulation will be pretty quick also. Okay, next slide. Okay, first look at cleavage, and this is going to be the cell splitting. The cells splitting, 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 dividing, 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 until we get the ball of cells. Okay. And here's just some patterns of splitting that you don't have to know these because we'll be going quick through these. But we start off with cells and two, and they double, they split, they split, they split. And this might be something like that's radially symmetrical, radial symmetry. Some worms, a sea anemone, um, sea urchins, starfish. Okay. In B, if you notice this cell's dividing and just taking on a slightly different orientation as they go. They might end up being spiraled a little bit. Okay, next one shows some more. Here we got two cells, four, a whole bunch. Oh, and notice this. It says it's left-handed coiling, and we end up with a snail shell or a mollusk. With this, here's some right-handed coiling in the next one, just slightly different arrangement of those, and we'll end up with that. Okay, so these are just different patterns of cleavage. Okay, the cells dividing, right? And like I said, we ended up with a ball of cells, one ball of cells. Okay, a whole bunch of cells in there. And now this next step, gastrulation is going to form three distinct layers, and all of our organ systems and organs develop from these layers. Okay, and it's a real big deal. Location, location, location. Where it's located, which layer of cell tissue it's in is going to depend on what that cell becomes. Because right now, before we start this, all the cells, they can be anything they want. We say they have a full set of DNA, a full set of 46 chromosomes. So we say they are unspecialized cells. Another word for that is going to be undifferentiated. And right now, that's the big hot topic with stem cell research. Okay, stem cells are unspecialized, undifferentiated. They can become anything they want to. In Alzheimer's patients, if stem cells are put in that section of the brain where cells are dying off, they develop some more brain cells. So wherever you put them, they have the DNA for it, and that's the section that gets read, and that's what they become. So location, location, location. So onward with gastrulation. There's going to be 3 distinct layers that we're going to need to see, and this looks pretty complicated. Well, don't read it all. Just pay attention to the 3 different colors on it. Okay. In higher animals, such as humans, we have 3 distinct layers. The outer part here is colored in blue. That is the ectoderm. The pinkish color, orange, salmon color, whatever, is mesoderm. And the inside, the yellow is going to be the endoderm. Okay. Endoderm eventually is going to form, oh look, the start of the gut. This is going to be digestive system. Okay. Blue, the ectoderm. Okay, this is going to be some of the skin tissues or integumentary system. We'll see this on the next slide in a minute. I think the mesoderm, I think muscle comes from that also. Okay, so like I said, location, location, location. All right, so gastrulation is formed during gastrulation. Form distinct layers, those 3 distinct layers. Okay, endoderm, ectoderm, mesoderm. Okay. Next slide. Okay, organogenesis. That's just a quick look at. We'll be stopping right after this slide. But notice we had the cells divided into cleavage. We got the 3 distinct layers forming. And then from these layers, endoderm, ectoderm, mesoderm, know those layers, we get all of your organs forming from that. Okay. And that is creation of those organs is organogenesis. Genesis, the first book of the Bible, means creation. So creating organs from those layers. And the last slide is shows what the systems that the ectoderm forms, the system the mesoderms form, the systems that the endoderm forms. So that is it. Thank you very much.